All right, welcome back, everybody. We're about to start our second oral presentation session. For this session, our first oral presenter is Effie Pereira, who works in the lab of Helena Ristic at McGill University. Her talk is entitled, Within Individual Oscillatory Patterns in Mind-Wandering and Attentive States are Associated with Functional Life Outcomes. So please welcome Effie.
Thank you very much. So broadly defined, we can think of mind wandering as attention that involuntarily shifts away from a task onto something unrelated, nicely depicted here by this Calvin and Hobbes cartoon of a mind quite literally running away from you. We've all experienced these lapses in attention that tend to occur across many different types of activities, such as commuting, when you're studying, or even when you're listening to a talk on a Friday afternoon. Given the ubiquitous nature of mind wandering, increased mind wandering has been found to have a fairly large impact on behavior. In terms of costs, increased mind wandering has been found to disrupt task performance in terms of reading and sustained attention. It's been found to interfere with processing, and it's also been found more commonly to result in depressed moods, such that more mind wandering is strongly associated with decreased levels of happiness. Conversely, mind wandering is also found to have beneficial effects on behavior. Increased mind wandering has been found to enhance creativity. It's been found to facilitate problem solving as well in terms of thinking of novel, outside-the-box ideas. And it's also been found to promote the management of long-term goals in terms of being able to prospectively think of yourself in terms of future. Given that we find these positive and negative outcomes to behavior within individuals, there's really a strong need to unpack the factors that drive these disparate positive and negative consequences that we see to behavior. Because as yet, we still don't have a really good answer to the question of how do we capture variability within mind wandering to be able to account for both these costs and benefits in behavior. Capturing variability in mind wandering has proven to be a little bit difficult within the field, typically because we tend to think of mind wandering in terms of aggregate terms. So more typically, if we were looking at mind wandering within a task, you might see something that looked at average proportion of mind wandering. So you're not really accounting for anything that's happening within the task, but you're specifically accounting for every single data point just based off of a participant. The problem with aggregating info in this way is that you really lose a lot of dynamic information that's happening within individuals and across the task. So for an example here, if you were to take one participant who scored at about 50% of mind wandering and look at what they actually did throughout the entire task. So if we were to ask them yes or no, were they mind wandering, and tabulate that, what we would find is that there's a lot of switching back and forth that ends up happening here. However, if we were to take another participant who had the exact same average mind wandering of 50%, we might find more stable patterns in behavior, such that you find these blocks of patterns over time, which you wouldn't really be able to uncover if you were just looking at this in terms of aggregate terms. So unpacking how mind wandering manifests in this manner should add a vital piece in being able to understand why we end up seeing these positive and negative aspects to behavior. Thus, using this method, my study was interested in examining whether we could characterize different patterns of mind wandering and attentive states within individuals, and see whether we could use these internal patterns to be able to predict both behavior and cognitive styles. So for our study, we recruited 100 participants here, and we estimated mind wandering using 3 commonly used tasks. The first was the experimenter caught method, where here we asked participants to read a book for about 60 minutes, and every 2 minutes we would interrupt them and simply ask them, were you mind wandering? Were you paying attention to your task? And they would continue doing this task until the full time had allowed. The second was a self-caught method, where again, we had participants read a specific book, except here we asked participants to specifically be aware of what they were doing while they were trying to focus on their task, so that they themselves would catch whether or not they were mind wandering as they were going through this. And they would continue through this until the time was up. With the third task, we gave participants what was known as a sustained attention task. Here, participants would be presented with a series of rapidly presented numbers, and they just had to continuously press a key over and over again unless they came to an infrequent target number where they had to withhold a response. So by being able to measure mind wandering across these 3 different tasks, we can capture not only a subjective measure of mind wandering and an objective measure, but also be able to look at this across different levels of awareness as well. So trying to make sure that we're capturing mind wandering that's fairly task invariant. In terms of measuring functional outcomes, here we gathered a wide range of stable behaviors. We measured the adult temperament questionnaire, which looks at individual differences in stable temperament traits that have been known to modulate with mind wandering. We looked at the autism quotient questionnaire, which looks at the degree of autistic-like traits within a typical population. And finally, we also collected transcripts so that we could get participants' most recent GPAs, because this has been known to be a fairly well-used marker of mind wandering as well. Taking all of these questionnaires and data, we conducted a factor analysis to be able to extract the latent factors that were most common across these behavioral and cognitive scales, which then extracted factors like attentional control, or the degree of attentional focusing that you might have for a task, your sensitivity to events, how social are you in terms of being able to project that to other people, and also academic achievement. In the interest of time, I'm only going to be focusing on 3 specific factors here, so that's going to be attentional control, academic achievement, and social skills. Now, in order to characterize the patterns of mind wandering that we found within each individual, we wanted to look at what mind wandering data looked like across the task. So similar to the example I presented earlier, we would just take one specific task, look at the time series across this, and just plot yes or no whether someone was mind wandering at any given point in time. We then use a moving window average to be able to convert this into a time series so that we can highlight some of the long-term trends within the data. We then use autocorrelations to be able to determine how periodic this signal actually was. So for any given example, autocorrelations would then end up repeating the signal, shifting it forward in time until it found where the correlation between these two were the strongest. So if you had a signal that was fairly repetitive, like this, you would find it to have a fairly high autocorrelation, and moreover, you would have this being a fairly high frequency because it's repeating quite often. But if, for example, you had a signal like this, where you really only had just a blip of mind wandering and nothing much more than that, you would find that this would have a fairly low to zero autocorrelation and no frequency. Once we characterized the within individual patterns of mind wandering, we used a cluster analysis to be able to differentiate and identify 3 distinct groups of mind wanderers that we had here, that's separated based on the different types of fluctuating pattern that they had within the data. So this is plotted here across the 2 dimensions that the cluster analysis extracted, and this is really done just to highlight the fact that these clusters did in fact separate quite well across the data. So our first cluster here, denoted in red, were the ones that we would find as the repetitive mind wanderers. So these were the ones that had fairly high autocorrelation. They repeated quite frequently in terms of their cyclical pattern. The ones in blue here, the ones that were the weakly repetitive mind wanderers, where again, they had a very strong pattern of repetitive mind wandering, but their frequency was a little bit less so. And finally, the third group here, denoted in purple, which we deemed as the irregular mind wanderers, where you can see that it really was just this kind of blip here or there, but they didn't really have any kind of significant pattern that we could really predict. Now, keep in mind these 3 different groupings here, because I will actually be presenting the results separated across each of these 3. Now, to examine the effect on real-life outcomes, all the graphs that I'll be presenting to you, as I mentioned, will be separated across each of these 3 different mind-wandering groups. So you've got repetitive in red, weakly repetitive in blue, and the irregular in purple. What you'll be looking at is proportion of mind-wandering on the x-axis, and this is just averaged across the entire task. And you're going to be looking at z-scores for every single factor that I want to present to you on the y-axis. So the first factor we'll be looking at here is attentional control. So what was the degree of attentional focusing that someone had within specific tasks or just within temperament traits? For the repetitive mind-wandering group here, we had a strongly negative relationship, such that more mind-wandering was associated with lower levels of attentional control. And the same negative pattern was found across the weakly repetitive mind-wandering group as well, whereas we found no relationship for those who had an irregular pattern. What this really highlights is that this consistent repetitive pattern that we see for mind wandering is really captured across how well someone is able to internally regulate their attentional control and attentional focusing. Moving on to the factor that looked at academic performance. Here we actually found differential results that separated our repetitive mind wandering groups. So for our repetitive mind wanderers, we found a negative relationship such that more mind wandering was associated with lower GPA. However, for our weakly repetitive mind wanderers, we actually found a positive relationship, where more mind wandering was associated with higher levels of GPA. This divergence really showcases how we can use these kind of predictive states or these predictive patterns to be able to associate these different kind of costs and benefits that we see with mind wandering. And similar to what I just showed you with the attentional measure with the irregular mind wanderers, we didn't really have much of a pattern here. Now moving on to the factor of social skills. Here we do in fact see that if we look at these repetitive groups, either strongly repetitive or weakly repetitive, we didn't really find any consistent relationship across these two. But with the irregular mind wanderers, we do find a positive relationship here, such that more mind wandering was associated with a greater ability to engage others in a more positive manner. We did find some interesting trends in the data with negative affect that I'm not going to be presenting here today, but I'm happy to talk about them as well later. So just to summarize, if we were to look again at the impact of mind wandering just on general behavioral outcomes, we can see that these costs and benefits that we find from mind wandering map on quite well to the different types of mind wandering patterns that we see, such that the costs of mind wandering are strongly associated with the deficits that we see for this repetitive pattern of mind wandering. The benefits that we see are strongly associated with the benefits that we see for the weakly repetitive group or the irregular mind wandering group. And it really suggests that these separations that we see across these different styles of mind wandering or different patterns of mind wandering do in fact uncover some relevant patterns that would be fairly invisible if we looked at aggregate data. So our data does in fact reveal that oscillatory patterns of mind wandering do in fact exist within individuals to slightly varying degree, that these internal patterns are in fact meaningfully related to individual differences in cognitive styles. And moreover, our study also presents some of the first findings into the underlying factors that result in these adaptive and maladaptive patterns of behavior across cognitive styles and behavioral styles as well. And it lends some good insight into what we might end up doing to target specific treatment for attentional deficits and for social deficits as well. So to end, I would like to thank my supervisor, Dr. Yelena Ristic, my collaborators on the project, Dr. Sylvain Baillet and Signe Sheldon, and thank you all for your attention.
Thank you for the talk. I have two questions, but firstly, I was wondering how you define mind wandering or how you explained it to your subjects.
So we primarily define mind wandering as instances where you would find yourself trying to focus on a task, but then you would find your mind either wandering away to something else. We specifically defined it in a rather vague manner because we didn't want to indicate that this was a positive or a negative thing to be able to push people to bias themselves one way or the other.
So I guess the reason I asked that is because of the distinction between task-related and unrelated, you could say. Like, are they mind wandering about the task or are mind wandering about totally something else completely?
Yeah, and that's a really good question. So for our two reading measures, so with experimenter caught and the self-caught method, not only did we ask them, were you mind wandering, we then asked them to kind of give a brief description of what they were mind wandering to and asked them, were you mind wandering to something that was related to the task or unrelated? This was also because we gave them a book, Sherlock Holmes, and sometimes people like to imagine what Sherlock Holmes would look like as he's going through these different types of activities. So we were able to filter out when it was mind wandering that was specifically not related to the task and only looked at that. I did also do a preliminary analysis, I think when it was at about 50 participants, to see whether there were any different trends in the data if we did separate across those, and we didn't find any differences there.
And my second question is, do you think, in terms of the consistency of mind wandering, you'd think perhaps maybe more consistently if it was more consistent if it's effectively charged, if it's, say, negatively valent strongly, and also if the content is consistent as well. And do you have anything in your data where you guys were able to look at that?
Sorry, could you repeat the first part of that? So you were talking about valence in terms of what people were thinking about or in terms of the task itself?
In terms of their thoughts, if you can see whether the content or effective valence is related to the consistency or lack of consistency in the patterns.
Yeah, I mean, I think that would be a really good question because there's a lot of interesting work that's looked specifically about whether you tend to focus more on negative thoughts as compared to positive thoughts and how that actually manifests across these. We haven't looked specifically anything in our data because we asked them to kind of classify their thoughts in terms of whether it was on task, off task, whether it was something related to them, something outside to others. So we wouldn't really be able to distinctly separate across that, but that would be super interesting for a follow-up study. So, especially because I kind of hinted at some of the differences that we're finding in the trends with negative affect that aren't coming out as strongly, but I wonder if that might be different. We'll talk later.
Thank you.
Thanks.
I just had a question about the task itself. You mentioned that you interrupt the people every 2 minutes.
Yes.
Don't you think that itself, the interruption every 2 minutes could affect their mind wandering because they're
Oh.
not able to concentrate.
No,
for-
absolutely. I think that is very much the case that could end up happening. It's one of the reasons why it tends to be one of the longer tasks, because we typically ask participants to do this for about 60 minutes, sometimes an hour and a half or so. I think there have been studies that have looked at whether or not interrupting them constantly might end up inducing mind wandering because they're now expecting the next interruption to happen there. It's one of the reasons why we didn't want to focus specifically on one task, and we looked at mind wandering across the 3 tasks to see if we could find differences across, say, a self-taught method or across a more objective method with that sustained attention. So it is possible that it may have done that. We didn't see any differences across mind wandering within each of these tasks. So the pattern of mind wandering for, say, one method was pretty consistent across the other methods as well. But yeah, there's definitely a ton of work to be done to see whether or not that task is in fact pretty great to use.
Thank you.
Thanks.
That's all the time we have for questions, unfortunately, but thank you, Effie.
Thank you.
So our next presenter is Samuel Burke-Nani, who works in the lab of Louis-Eric Trudeau at the University of Montreal. His talk is entitled Cell Loss in Parkinson's Disease. To foresee, prevent, and treat, we must understand where, when, and why. So please welcome Samuel.
Excuse me. So Parkinson's disease is a degenerative disease characterized by progressive degeneration of dopamine neurons of substantia nigra. And this degeneration is thought to contribute to the major symptoms which most of us are probably aware of, which are the motor dysfunctions which we see in PD. But one of the things which is notable in PD is that it's not just a motor disorder. And what you can see in this figure here is this is just a description of the broad different symptoms which exist in the disease. And what I think is important to bear in mind is I don't think all of these, and the field doesn't think all of these symptoms can be attributed to the dysfunctional degeneration of only dopamine neurons of substantia nigra. Within the pathological data in the PD field, there's 2 main well-documented phenomena, and that is something that's called Braak staging. And so this is the aggregation of alpha-synuclein, and then there's cell loss, which are subtly different but independent phenomena. And if we know, if any of you are familiar with this, Braak staging is a way to describe the progression of the Lewy pathology in a Parkinsonian brain. And that is that you have deposition of these Lewy bodies beginning in the brainstem. And as a function of disease progression, we see these spreading up the brainstem and into the majority of the cortex. Cell loss data, on the other hand, shows a similar pattern, though subtly different. And here what there is this canonical notion that this degeneration does begin within the substantia nigra, but there is also degeneration, so cell loss within other regions, notably of the pedunculopontine nucleus, of the locus coeruleus, the raphe nuclei, the dorsal motor nucleus, the vagus. So one of the key questions in the field is, why are these neurons degenerating? Why are they dysfunctioning? And I think to understand why, we have to know where, because it's important to compare these 2 things. If you have a feature which you determine is causing degeneration of a single neuronal subtype in PD, it should be also what is responsible for degeneration in other subtypes. So that is kind of where I'm going with this talk and the perspective at which I'm looking at PD in our lab. A key concept in PD and understanding the causes of the neurodegeneration is this idea of selective vulnerability. And that is, what is it about these neurons that distinguishes them from other ones and renders them at risk to degeneration and renders them, well, to degenerate over time? And what we know from the literature so far is there's a strong association with age being the biggest risk factor with PD, but also there's many genes associated, which I won't be discussing. We heard Tyler this morning talking about some of those. We have environmental factors. There's a whole broad group of individuals who are looking at alpha-synuclein. But what I'm interested in, is really the autonomous properties of these neurons and how that might explain why they're degenerating. And this includes, for example, their nature. They're autonomous pacemaking cells, and they rely on calcium mechanisms for this autonomous pacemaking. But what I'm really interested in is their other features, which is their axonal arborizations and the number of neurotransmitter release sites. And these all should together, or the idea is that these are contributing to a very high bioenergetic burden on these neurons. And over a lifetime, this is what is resulting in elevated mitochondrial oxidative stress, resulting in subsequent degeneration. In our lab in 2015, we published a paper demonstrating a causal link between axonal arborization and oxygen consumption, and then subsequent vulnerability to a body of stress assays. And what was interesting here is we're able to manipulate the growth of dopamine neurons in substantia nigra, and with that manipulation, you're able to change their relative vulnerability to multiple different stress assays. So as these neurons become more arborized, or as you decrease the arborization of these dopamine neurons, they're less vulnerable to certain toxins. And so our work is moving forward to try to see whether this hypothesis carries forward into these other groups. One thing that I mentioned was that there's canonical notions of cell loss and which regions of these brains are degenerating. And something that became apparent to me and a student in the lab was that when we looked at these papers claiming cell loss was occurring in certain regions, we'd look at the cited papers and you'd be surprised that, well, they didn't actually quite show that. And so this motivated us to do a systematic review where we tried to find all these papers documenting cell loss in Parkinsonian brains. And what I have here is a summary of what we think is probably what the literature shows, and that is to show that yes, there's definitely degeneration into substantia nigra. There's degeneration in the VTA as well. Regions such as the Raphi nuclei are said to be degenerating. However, what we noticed was the only study using stereology, which is the gold standard method of counting cells in a brain sample, actually showed no loss in these regions. And for the locus coeruleus, for example, another brain region said to be degenerating PD, there's not a single study using stereology to quantify cell loss in that region. So this is to say that not to hold these assumptions to the models, and as I'm moving forwards and trying to test this hypothesis, to hold that in mind. And so what I'm currently working on is trying to look at the relative vulnerability of PD nuclei to a body of stress assays as a function of axonal arborization, the number of varicosities, so neurotransmitter release sites, and I'm looking at other bioenergetic variables such as the density of mitochondria, their relative production of reactive oxygen species. And just to give a little snapshot of what we're doing is we have a primary neuronal cell culture system from mice, and what we do is we grow these neurons over a week and then quantify their axonal arborization and describe what these cells actually look like in vitro. And from that, we can get ideas of how close are we to replicating this phenomena of massive axonal arborizations. And so what you can see here on the left-hand side is just a panel showing how we actually do the quantification. And what you can see at the top is we can see between the SNC and the VTA, there's this markable difference in axonal arborization. And when you look at neurons of the locus coeruleus, they have these massive axonal arborizations, which is also found in vivo. What I see here in vitro is I'm getting neurons which have arborizations of somewhere in the region of 3 to 5 to 10 centimeters. A mouse in vivo is somewhere in the region of, say, 10 to 50 to 60 centimeters. And in human, these neurons are thought to be in the region of meters. So this idea, I think, is key to trying to understand how does this energetic burden to support the axon confer or contribute to their vulnerability to stresses associated with PD. A second thing we're looking at in our in vitro system is describing the varicosities. Varicosities are neurotransmitter release sites. They're going to cost energy to maintain and to use. And what we find, even in vitro, is that we have varicosities present along these axons, that these neurons are able to release neurotransmitter all along their axons. And then finally, within the 10 minutes I had, I wanted to show some first data that we're doing and what I hope to be doing in the future. But it's just doing simple stress assays on these neurons in vitro to be able to compare their relative vulnerability to PD-relevant toxins. And so just quickly, what I'm showing you here is just a simple hydrogen peroxide stress assay where we challenge these cells to hydrogen peroxide and then quantify how many cells survive. And what we see, briefly say at 150 micromolars, is the emergence of this differential vulnerability between substantia nigra dopamine neurons and VTA dopamines. But what was funny, or was striking so far, is despite the fact that I've seen locus coeruleus neurons have such vast arborizations, they seem remarkably resilient to hydrogen peroxide stress assay. As I move forwards, I'm going to be doing these tests in that whole plethora of groups, so looking at cholinergic neurons as well as projecting neurons that are not thought to degenerate in PD, and then looking at multiple other stress assays to compare the different mechanisms that could be contributing to the degeneration. And I'd just like to thank predominantly the group that I work with, the people I'm working with, and also the funding agencies and FRQS for giving me money. So thank you very much, and thanks for listening.
Hello.
Hi.
Hi. So I just had a question because you do all of your work, a lot of work in vitro.
Mhm.
Is there a reason for that you don't look directly in the brain of the mice for morphology? Because morphology can be quite different in vitro and in vivo. So.
Yeah. So in our lab, we do work in mouse in vivo, but obviously technically it's more difficult to, say, quantify axonal arborizations, not just because you have to get the brain, but then how do you actually trace an image? So a lot of the basic stuff, so quantifying arborization, seeing how that affects its vulnerability to a simple assay, I think is a good way to pass whether that is a factor which is contributing to their vulnerability. And secondly, the plans are we do this stuff in vivo. One of the other students, he's been manipulating the D2 receptor and dopamine neurons and has found that regulates the arborization of these neurons and then regulates somewhat their relative vulnerability to 6-HDA injections. And if you're trying to look at mitochondria as well, it's much easier in vitro than it is in vivo, so that'd be one reason.
Hi, thank you for such a nice talk. I was wondering about the cultured neurons you showed at the end. I guess you differentiate the SNCA from the VTA neurons in the dissection and the dissociation, but how's the morphology when you're growing them? Could you stain them with any markers or tell them apart visually?
So distinguishing between dopamine neurons is quite challenging because they're similar in a lot of ways. And I was actually having this conversation recently. Can't we culture them together and then just do an immuno to distinguish between the two? But as far as I'm aware, I don't think there is a reliable marker unless you can tell me that there is.
Not that I know of, no.
No. But what I can say is the VTA, the mesencephalon can be divided into about 5 different subpopulations of neurons, as Poulain published a paper in Cell a couple of years ago. And we did some fact sorting experiments on our dissection. And so for our dopamine SNC dissection, we really take the most lateral wings of the mesencephalon, and the VTA, we take the central region, leaving out the like in-between area. And what we found is in the RNA-seq data that these neurons really do look like those 2 different sections as described by Poulain's paper. So that's how I say that this is a SNC neuron, because we did the dissection. Does that answer? Yeah.
Yeah, that answers that. Maybe another similar quick question. That like very long, elaborate axonal arborization, how long do you have to culture the neurons just to see that form?
So for the tracing which I showed, which is this is just 7 days. It's incredible how much they grow. And so I did like a rough calculation. I think I see about 60 to 70 micrometers, I think, an hour of, I'm forgetting. Anyway, they're about 10,000 micrometers long after 7 days. And perhaps if we can keep them going for many weeks, they might get even bigger. But what I think the most interesting thing is when you look at a dopamine neuron, it projects from the mesencephalon, and it's maybe a single axon until it reaches its target area, whether it's the accumbens or the striatum. For the VTA, the accumbens. But what we see is we have that branching pattern before it's even, say, reached its target. So were they to have that growth and then do the branching, I'm not sure in vitro how far we could go with our arbors.
Thank you, Samuel.
Yeah.
Our next presenter is Blondine Pomeroy, who works in the lab of Dr. Keith Murray at McGill University. Her talk is entitled, Human iPSC-derived Down syndrome astrocytes display genome-wide perturbations in gene expression and an altered adhesion profile. So please welcome Blondine.
Thank you. Good afternoon, everyone. So today I'm going to be telling you about my PhD project, which is, as we just heard, on human iPSC-derived astrocytes in the context of Down syndrome. So for those of you who are not super familiar with Down syndrome, it's caused by the triplication of chromosome 21, and it's the most common genetic cause of intellectual disability. It affects the entire body, but considering that we study the brain, I'll be focusing on that. So in the brain, they've been shown to have a smaller brain volume, and that's been mostly shown to be caused by a lower number of neurons and synapses. And as you can see over here on the control dendrite, you can see lots of little spines that you would expect to have, whereas in a Down syndrome individual, there's a lot fewer spines, and they're pretty abnormally shaped and dysmorphic. Interestingly, also Down syndrome increases the risk for a lot of other brain diseases such as Alzheimer's disease, autism, and epilepsy. So it's a pretty profound neurological disorder. But as I mentioned, I study astrocytes and not neurons. So astrocytes are the most common type of glial cell in the brain, and they play a lot of variety of different roles. One of their roles is to provide structural and metabolic support to the brain and neurons. They also maintain ion and neurotransmitter homeostasis, and they're altered in almost all brain injury and diseases, which makes them a pretty huge untapped therapeutic potential. So in Down syndrome, there's relatively few studies in comparison to neurons, but we do know that there is an altered astrocyte support to neurons, and that they have an increased oxidative stress, and that they have a reduced secretion of synaptogenic molecules. So I personally find that really interesting because it's very likely that a lot of the neuronal pathology that we see in Down syndrome is actually caused by defects or alterations that are in the astrocytes and not necessarily the neurons. So for my project, I wanted to study human astrocytes in particular, and so for this I've used induced pluripotent stem cells. And just to introduce that briefly, this is a relatively new technology which allows you to take an adult differentiated cell, like the fibroblast in my case, and insert 4 Yamanaka factors, which allows for their reprogramming into an induced pluripotent stem cell. And from that, in theory, you can then differentiate them into any cell of the body, and in our case, of course, we're interested in the astrocytes. So this is essentially what I did in the first part of my PhD, and I obtained 6 cell lines, 3 control and then 3 Down syndrome fibroblast cell lines that I differentiated into induced pluripotent stem cells, and then differentiated those into NPCs and then astrocytes. And those are some immunohistochemistries of each step. You can see on the far left some of the iPS cells, then the NPCs, and finally the astrocytes. So after having generated those cells and characterizing them, we decided to perform RNA sequencing on the astrocytes in order to get a pretty overview of what has changed in Down syndrome astrocytes, and that was also because there was very little known on those cells. So that's what we did, and this is some of the data that we obtained. So here you can see a Manhattan plot where on the bottom is the chromosome number, and those green dots are all of the upregulated genes that we see in the Down syndrome astrocytes, and the magenta ones are the ones that are downregulated. So overall, we saw a really global transcriptome deregulation. You can see that every single chromosome have differentially expressed genes that are both up and down regulated, and it's not just the chromosome 21 that's affected. Interestingly, though, if you do look at the chromosome 21, you can see a major skew towards the upregulation of genes on that chromosome, which is to be expected because you have an additional chromosome. But what was really interesting to see for us is that it's a really global transcriptome misregulation that we observe. So going from there, we wanted to see what biological processes were affected and what all these changes in gene expression actually meant biologically. And so for this, I performed a gene ontology, and this showed, quite interestingly, that a lot of genes related to cell adhesion and extracellular matrix were misregulated in the Down syndrome astrocytes, and all of those were found to be in the extracellular matrix component of the cells. So this was quite exciting for us because that's not something that's ever been reported in Down syndrome astrocytes. So then I went on to validate all of those genes by qPCR to ensure that those changes were indeed real. And then we decided to perform another sort of global experiment to look at the overall state of these cells, since again, not very much is known. And for this, we decided to look at the chromatin state of the Down syndrome astrocytes, and we used a method called ATAC-seq, which for those of you who don't know this method, it basically enables the insertion of a transposase, which cleaves the open parts of a chromatin, which are then purified and sequenced. So essentially, it allows for the sequencing of all of the open chromatin areas of the cell. And interestingly, we got very similar results to the ATAC-seq, to the RNA-seq, where we saw very global genome-wide changes. Here again is a very similar Manhattan plot where you can see all of the chromosomes on the bottom, and you can see in this case we're looking at chromatin organization, and we can see that in every single chromosome there's some sites of chromatin that are more open and less open. So again, the addition of the extra chromosome is causing global changes. And I performed gene ontology again to look at what biological processes were affected, and really interestingly and neatly for us, the same processes showed up to do with extracellular matrix and cell adhesion, and they're all situated in the cell, same components of the cell. So both of these very global experiments both hinted us in the direction that the cell adhesive state of Down syndrome astrocytes is most likely altered. So I then went on to perform more functional assays to look at their adhesive properties directly. And the first thing I did was to look at their size, because that's actually something that I'd noticed in culture, that the Down syndrome cells maybe looked a little larger. So I did some immunohistochemistry and validated this observation that the Down syndrome cells are indeed larger than the controls. And this is only found in the overall cell size and not their nuclear size. And we then did a flow cytometry experiment where we did the same thing, but we looked at the size of the cells in suspension. And in that experiment, we saw no difference, which was really quite interesting because it means that the cells themselves are actually bigger. It's only when they're spreading on the dish that they're larger. So it's more of an increased cell spreading than actual increase in size, which makes a lot of sense when we saw all of these changes to do with cell adhesion and ECM. So another cell adhesion assay that I performed was to put the cells in a petri dish, which is an uncoated plastic dish. And in this context, they're supposedly going to adhere to each other rather than the dish, since it's not coated. And they're supposed to form these little aggregates that you can see over here. And what we saw in this case is that the Down syndrome cells formed much smaller aggregates, and they're less able to adhere to each other. But really interestingly, they're not just floating in suspension, unable to adhere to each other. They actually just adhere to the dish itself, which is not really something that you would expect from the cells to do, since it's just an uncoated petri dish. So what this experiment showed is that the Down syndrome astrocytes have an altered adhesive profile, and they have a preference to adhere to a substrate rather than to themselves. And finally, I did another adhesion assay in which we looked at a family of proteins specifically that we saw were downregulated in our RNA-seq experiment, and those are protocoderins, and they are very important for homophilic cell adhesion. And first of all, we confirmed through Western blots that they were also downregulated at the protein level. And once we'd done that, I did an experiment in which I coated some cover slips with 3 different protocoderins, 2 of which hadn't shown any changes in the Down syndrome, and 1 of them was shown to be downregulated. And what you can see here in the control condition is that in all of the different protocoderin-coded conditions, there's more cells that adhere to those than to the untreated glass. However, in the DS, there's only an increase in adhesion to the 2 protocoderone-coded conditions that had no change in gene expression, but there's actually a deficit in the one that did have a decrease in expression. So overall here we showed that DS astrocytes have a specific deficit to protocoderone gamma-mediated cell adhesion. So overall, if I haven't lost you, I've showed that I've demonstrated that there's a whole transcriptome deregulation found in Down syndrome astrocytes, and that the differential gene expression affects the extracellular matrix and cell adhesion. We've also found genome-wide changes in the chromatin accessibility of these cells, and again, the differential promoter accessibility affects the ECM and cell adhesion. And finally, I've shown that there is a functional change in the adhesive property of these cells, and they have an increased cell spreading, a decreased cell-to-cell adhesion, increased cell substrate adhesion, as well as a deficit in protocoderone gamma-mediated cell adhesion. So with that, I'd like to thank my lab and my PI, as well as the collaborators, all the funding, and you for your attention. Thank you.
Hello. Thanks for the talk. That was really interesting. So I was just wondering, so you saw changes in both the genomics and also the transcriptomes, right?
Yes.
I was wondering if you've correlated the two to see whether the more accessible genes are the ones that are upregulated in the transcriptome. I know this is expected, but it doesn't have to be.
we did actually, and there's not a great correlation, to be honest. And that's something that other groups have reported, specifically in DS as well, but in for other diseases and conditions as well. And there's lots of reasons why they may not be directly correlated, because the epigenetics and gene expression don't always correlate. But in our case, we would have expected it to be a little better than what we actually found. But the fact that we still found ECM and cell adhesion in both of these experiments was reassuring, in the sense that we were pretty confident that we were going to see some kind of biological functional phenotype, but it was a little surprising not to see a great correlation.
Just a follow-up thing. It might be really cool then if you look at the proteomics as well, just to see what happens on the protein level on the translation.
I'm actually doing that right now.
That's great.
Yeah,
I look forward to that data.
Yeah, I'm looking at both the whole cell proteomics as well as just the extracellular. Like, I've isolated the extracellular matrix, and we're doing proteomics on that as well. So that'll be interesting. Thanks.
Hi, really good talk. Thank you so much.
you.
So you mentioned in the earlier slides that you think that maybe it's the astrocytes that are affecting the neurons and not necessarily the intrinsic abilities of the neurons.
Mm-hmm.
Have you thought of maybe doing an experiment where you have the co-cultures of astrocytes and then have wild-type neurons and see and measure dendritic spines afterwards and see what happens?
Absolutely. That's been that was actually my primary experiment that I wanted to do when I first started, and then I realized that it was actually quite hard to do that, especially with iPS cells. But one experiment that I'm actually hoping to do relatively soon is, I just mentioned that I've isolated the extracellular matrix to do proteomics, and I was thinking of growing neurons on top of just the ECM on its own, since that's what we've seen in our cells is mostly changed, and that might actually also just technically be easier than doing co-cultures. Because I've tried, but with very moderate success, to be honest. So.
It's really impressive work.
Thank you. Thank you.
So our next presenter is Cassandra Lowe, who works in the lab of J. Bruce Morden at Western University. Her talk is entitled Prefrontal Cortical Activity Modulates Susceptibility to Obesogenic Eating Behaviors. So please welcome Cassandra.
All right, great. Thanks so much for having me here today. I'm really excited to talk about my work that has looked at the role the prefrontal cortex plays in modulating obesogenic eating behaviors. And by that, I mean those excessive consumption of those foods that are high in fat, high in sugar, really calorie dense, and ultra processed. So if you're here this morning, you would have realized that these diets are really, really, really bad for our health. Not only are they the leading cause of chronic disease, including a pivotal risk factor for the development of obesity, they can have really negative impacts on our brain health. The neural inflammation associated with obesity can actually increase our risk of developing things like Alzheimer's and Parkinson's disease. So, moral of the story is, when we leave here today, don't have any of those cookies outside there. But it's a lot easier said than done because we do live in an environment that really promotes overconsumption, in the sense that we are always exposed to food cues. Everywhere we go, there's these cues to eat these really palatable, really delicious foods. And they're never cues for things like apples, carrots, and kale. It is very specific to these calorie-dense foods. And they are extremely cheap to get, especially when you're eating out. They're often a lot cheaper than things like a salad or some vegetables. So because of this, some individuals find it really hard to regulate how much of these foods they're eating. And a lot of my research has centered on why. Why are some individuals better at this than others? And more specifically, can we develop a model that can be used to explain what's going on at a neurocognitive level that can explain why some people are more vulnerable to overeating than others? And then can we target this to improve dietary habits? And when we go back to this idea of the modern environment, we do live in an environment that really depends on us to control this automatic tendency to want to eat these really hyper-palatable, super delicious foods. And this control abilities have been linked to the prefrontal cortex and our executive functions. So my work has really centered on, can individual differences in prefrontal activity explain why some people are more vulnerable to overeating than others? And to address this question, I have used repetitive transcranial magnetic stimulation methods, specifically continuous beta burst stimulation, which is a way of decreasing activity within specific regions of the cortex, to examine what goes on when we decrease activity in the dorsolateral prefrontal cortex. So for the purpose of this talk, I'm just going to present findings from 2 of my studies. I just combined them together so you can get an overall idea of what's going on. So what we see is when we decrease activity in the left dorsolateral prefrontal cortex, there's a significant reduction in cognitive control, which in this case, an increase in Stroop and flanker interference scores. So higher scores are indicative of poor performance on these tasks. And the violin plots here just kind of show you the distribution of this. And when we look at how much people eat during our bogus taste test, and by bogus I mean we're telling people they're doing a taste test, but we've really measured how much they're eating. We do see a significant increase in how much people are eating when we decrease activity in this brain region, but it's very specific to the foods that people find the most appealing. So in this case, it was chocolate and chips in comparison to plain rice cakes or unsalted soda crackers. And what is even more interesting is when you look at the individual changes in how much people are eating of these really appealing foods, you see an increase in almost 82% of people, and these were all mostly normal weight, healthy individuals. But what is even more interesting is there's substantial variability in this effect, where some people show these really large increases in how much they're eating, where other people show really small changes or none, or a decrease. And what I think is really interesting going forward is figuring out why. Why are some people showing these really big differences and other people are not? I don't have an answer for it. I just do think it is a really interesting question to go forward. And then in one of the studies, I did do EEG measurement of intentional engagement to high-calorie and low-calorie food pictures. And what you see is there's a significant increase in this periphery A amplitude specific to high-calorie foods following CTPS. This means that we're spending more attention towards these foods. We have lower cognitive control abilities, and I didn't put this data up here, but participants did report stronger food cravings for these really appealing foods. But when you consider all these variables in tandem, it is this the reduction in cognitive control abilities that is driving this overconsumption behaviors. So I did just throw a bunch of information at you. So what does this mean? It means that the functionality of our dorsolateral prefrontal cortex does play a causal role in overeating behaviors, and it is driven specifically by changes in our cognitive control abilities. So the next focus, and this is really truly the heart and soul of where I want to go in the future, is can we improve prefrontal function to improve dietary choices at a large-scale level. And the reason why I'm particularly interested in this is if you were here this morning, you would have heard my wonderful colleagues talk on how these diets really do have a negative impact on our brain. And in our recent Trends in Cognitive Science paper, we have outlined kind of this vicious cycle where you have these brain characteristics that can lead to this overconsumption behaviors that further leads to dysfunction and other things in the brain. So can we take these early preventative measures to offset this cycle? Can we prevent the cycle from going forward? And when we think of preventative measures, one of the most promising out there is exercise. So not only is exercise one of the best things you can do for your health, there's substantial evidence out there showing that it does really have a beneficial effect on both prefrontal structure as well as white matter tract integrity, and as well as the hippocampus, which is another reason often associated with overeating-like behaviors. And you do see that it has a substantial impact on our cognitive control abilities. And some work that I've also done has shown that it can be used to both offset and promote recovery from CTPS-induced reductions in prefrontal activity. So when we do exercise before applying CTPS, you can see here that there is no change in this, in the flanker interference scores in this case. And also when you take CTPS or do exercise after CTPS, it helps promote a recovery. So people recovered faster from the CTPS. So I didn't show it here, but we did have control exercise conditions. And so why am I showing you this data? Well, I think it's really interesting in the sense that there are a lot of factors out there that can actually influence our cognitive abilities. And there are things that are happening to everyone, such as being a little bit stressed, not getting enough sleep. And what these data show is that maybe exercise might be a way of kind of offsetting these things. And then in turn, it might actually help us make better dietary choices. We often hear about stress eating and stuff like that. So maybe we can use exercise as a way of offsetting that impact. And when we look at the actual effects of moderate intensity exercise, we do see this graph, you can't really see it very well, but in comparison to very light conditions, there is a significant increase in cognitive control ability, so there's lower interference scores, but when we actually looked at how much people ate in the sim taste test, we do see a significant reduction, again, in the repetitive foods that they're eating following this moderate intensity exercise. And this was mediated by changes, the exercise-induced changes in executive functions or cognitive control. So I have two main take-home messages that I want to leave you with today. So first off is that vulnerability to overconsumption may be attributed to individual differences in prefrontal activity and cognitive control. So this gives us a model that we can use to kind of understand why some people may be more likely to overeat in this modern food-rich environment. And the second take-home message is, so my absolute favorite saying is, you can't outrun your fork. And that gets at the fact that a lot of people will use exercise as a way of offsetting calories they've consumed. But instead, maybe we should think about how we can use exercise as a way to improve people's dietary choices through increased cognitive control and things like that. So, thank you very much for listening to my talk, and thanks. Yeah. It's okay, I can hear you okay. Oh, sorry. Yes. Oh, it's okay. Okay, I'm really great that you asked that. So one of the things I'm really interested in going forward is testing the notion that we can in long-term exercise interventions. So there's quite a few out there that have shown that after randomized control interventions for different periods of time, but people do see a significant increase in cognitive control abilities. This is observed mostly, a lot of the research is in older adults, but there's some in children as well. So it does help improve cognitive abilities. What's not known right now is how long those effects last. I think that'd be really interesting going forward, but there is enough out there that's showing that these randomized trials do that. In terms of whether high intensity exercise versus the moderate intensity. So the reason I chose the moderate intensity is I wanted people to participate who wouldn't necessarily super active. And high intensity tends to kind of scare people off. Where the exercise protocol we used, we just made people walk on a treadmill and we adjusted the incline, so it was something everyone can do. There is a lot of research that has shown that high intensity exercise does have a really beneficial impact. I find that a lot of that work, the participants tend to be very fit to begin with. And the one thing that's in that literature that still needs to be worked out is what do we consider high intensity interval training? Because it seems like every study has different intervals. And the main reason I also didn't use that is that with high intensity exercise, you actually see a decrease in the acute effects, where you do see more long-term stable effects in executive functions. Does that answer your questions, or thanks?
I don't know. Hi.
Hi.
I was wondering for the eating task where you were checking how many calories they'd eat for the appetizing food. How could you control for like just some participants being more hungry? Like, wouldn't that really like increase the variability of your data if someone's like really hungry and he's having lunch in front of you?
Yes, so with all the studies we used within subject design, but we asked people to eat 3 hours before they came in, and we kept it all in the afternoon, so there was very set times. We also measured how hungry they were. I didn't present that data, but across studies, there was no significant difference in like self-reported hunger. We also measured like when they last ate, and there was no differences that as well. So it wasn't directly controlled, but I tried to control for it as best as possible.
Okay, yeah, that's good. Thank you.
Oh, I think there was one more. Sorry, is that okay? Or? Okay, thank you.
Our last presenter for this session is Ricardo Pizarro, who works in the lab of Amir Shmuel at McGill University in the M&I. His talk is entitled, Using Deep Learning Algorithms for Automatic Quality Control of Brain MRI. Please welcome Ricardo.
Hi everyone, my name is Ricardo, and today I'll be describing 2 projects that I've been working on where we develop deep learning algorithms to make a specific prediction about MRI data. So before we get into the specifics of the projects, I wanted to just introduce some concepts about deep learning to give you a sense for the type of work that we do. So deep learning, we can think of it as a subfield of machine learning, which in itself is a subfield of artificial intelligence. And AI is the general concept of trying to mimic human intelligence. So then in machine learning, what we try to do is to develop an algorithm that can make an internal representation of your input data and then make a prediction about something in the future. So the algorithm is learning if the prediction errors is getting reduced. So machine learning is very powerful and can be used for detecting spam emails or for detecting objects in an image. So for example, let's say you're interested in detecting this stop sign. The programmer might identify some features that they'll combine in the end. So for example, one of the features might be the letter S, or the color red could be very useful, or the octagon shape. And at the end of the day, what the algorithm does is it'll combine these different features and then make a prediction saying, yes, this is a stop sign, or no, this is a picture of my mom. So then in machine learning, what people did is they introduced additional layers to increase the complexity of the algorithm. So here I'm showing just a very similar architecture, but we have an additional layer here, layer 2. And with this additional layer, what we can do is then take the input and combine them in different ways. And then in the second layer, we then combine it again in a different way. So this increases the level of sophistication, the complexity of the algorithm. And that's what deep learning did, is they built on this concept of adding layers. And so they added more and more layers, and then you get this deep learning. So this is one example of deep learning, of an architecture that's used in deep learning. This is called the AlexNet. And you can see it's very complex. It has a lot of layers. And this is a convolutional neural network. So some of the key features of this architecture is that it uses convolution, max pooling, and a rectified linear unit. So with these three mathematical procedures or processes, this architecture was developed to very accurately detect the content of images with very high accuracy. So another key difference between deep learning and machine learning is that the architectures are very complex. This specific architecture is made up of 1 million parameters, and so you need a lot of data to train it well. So typically you want tens of thousands of samples or hundreds of thousands of samples in order to properly train it. And this is where we come into neuroscience. So recently in neuroscience, there's been a trend of these neuroimaging databases emerging where the people are trying to increase the power of the study by acquiring more and more data to study a specific disease or study the human brain. So these neuroimaging databases are acquiring lots and lots of data. They have up to tens of thousands, hundreds of thousands of data sets of brain imaging. And so this is exactly what we did in the first project. We developed a deep learning algorithm to do something very simple, which is to detect the contrast of an MRI data set. So this work is in collaboration with NeuroRx and was published in Neuroinformatics last year. So NeuroRx is a company that's contracted by pharmaceutical companies to process the MRI data set. So NeuroRx will receive hundreds of thousands of scans from different neuroimaging sites across the world, and their job is to process the data set and objectively report some metrics, some results to the pharmaceutical companies. And then so for our goal here was to detect the MRI contrast, and a lot of times NeuroRx will receive data from neuroimaging sites that's incorrectly labeled. So when they receive incorrectly labeled data, then it messes up the analysis, you get wrong results, and everything goes wrong. So for those of you who are not familiar what MRI contrast is, this is for part of the study, this is what we did. We wanted to identify these 5 different contrasts. So MRI can be used to image the brain, and by programming it in a different way, you can highlight different tissues accordingly. So for example, if you just focus on the bottom 2 images here, the proton density and the T2, you can see that different parts of the brain are showing up differently in the 2 images. And at the top here is the entire data set that we used. We had around 40,000 MRI data sets, and we divided into a training, validation, and testing data set. The training we used to optimize our parameters of the algorithm. The validation we used to assess the performance while we were training. And the testing we kept out completely separately so we can assess the performance of our algorithm at the end of the day. And so this is the architecture that we used for our study. It's very similar to the one I showed you before, the AlexNet. And so what I wanted to show here is that the input to the algorithm is a single slice of the MRI volume. And the data goes into the architecture, and the output of the architecture is a probability that this particular slice belongs to a particular category, in this case, T1 before contrast. And then in a similar way, we can do the same thing with other slices, proton density here. And then at the end of the day, we summarize all these results in this confusion matrix. So on the y-axis is the prediction as done by our algorithm, deep learning, and on the x-axis is the ground truth, the target modality. So if we look at the diagonal of the matrix, we see where the two are in agreement, and off diagonal is where the two are in disagreement. So we can summarize everything with the accuracy, which I'm showing up there. And so great. So next, what we did is we adapted this architecture to now detect the artifacts in MRI. So this is a study that's ongoing still. We're still working, and it's in collaboration with NeuroX. And now we are, instead of identifying the contrast, we want to identify the artifacts. So in a similar way as what I described earlier, if you have MR images that have artifacts in them, it can lead to type 1 error, it can lead to spurious results in the processing pipeline, it might detect a lesion that's not there. So in the study, what we did is we identified 3 different artifacts as defined using the standard by Loris. So I'm showing here just 3 images to illustrate what kind of artifact we might have. So the one that's labeled intensity up there is a signal dropout, possibly due to metallic implants. The motion is when the subject being scanned is moving around in the scanner. And then the coverage one is where the MR technician might have incorrectly placed the field of view, so then the top of the head or the cerebellum might get cut off. Another thing you might notice is that the number of data sets that we use here is much smaller than in the previous study, because we had one of our colleagues manually label each of the data sets. So we divided again in the training, validation, and testing. And I'll just go quickly because it's very similar to what I showed before, but you have now our architecture is working in 3D. So rather than feeding in a single slice, we are feeding the entire volume of the MRI. And again, the output is a probability that this MRI volume belongs to a specific category. So this case is identifying this volume as a intensity artifact or a motion artifact, and then this one as a coverage artifact. And again, we summarize the results with on the testing data set. And what we see is that, thank you, with this algorithm, we can, and on this data set, on this testing data set, we can detect whether or not an MRI has artifact with 100% accuracy, which is great. So if we take this to other databases and we're able to use this data set, this algorithm, to just filter out any problematic MRI volumes, then we can then reduce the number of volumes that we need to inspect. So if, however, if we break down this number 107 into the different categories that we were aiming to classify, we see that there's still room for improvement. So I just wanted to end by reiterating that deep learning is a very powerful tool that can be used. In this case, I talked about neuroimaging databases, but it can be used in different studies, with different data sets. I've described 2 studies that we've been working on where where we detected the contrast of the MRI, and another one where that's still ongoing, but we're looking to identify whether or not an MRI has artifacts and what type of artifacts. And so if you're interested in this type of work, I just wanted to let you know about this group that Arna Ghosh and I have recently started, where we're trying to learn how neuroscience and deep learning can help each other. So these artificial neural networks are biologically inspired neural networks. They're not exactly what's going on in the brain, but they're modeled after that. And so deep learning has a base in neuroscience. And now that deep learning has been developed, it's being applied, as I've showed you, in neuroscience. And now there's even more work where more advanced or more detailed neuroscience research is helping to develop deep learning even further. So if you're interested, check this out. And with that, I want to thank my collaborators, Setu, for helping me with the image artifact detection, and the collaborators at NeuroX. And I wanted to thank you for your attention.
Thank you. Very interesting. I wanted to ask actually about your last comment, this intersection between neuroscience and deep learning, in terms of to what extent deep learning does reflect a biological mechanism. I think in the field there's some kind of argument. So for example, you need a huge number of exemplars, and some people claim that when our brain develops, we actually develop this model without so many examples, and also that you know, some people blame deep learning as like just a stupid overfitting model, you know. So I wonder just like what are your thoughts about that debate, and where does it stand?
I see some parallels between the two. For example, in the architecture that we use, this convolutional neural network, the way it's processing the data, there's a lot of parallels. I don't want to say it's similar, but there's a lot of parallels how the data is processed along the different layers. So, for example, in the first layers, the architecture will look for these edges. Like edges are very excite the artificial neurons quite a bit, and then as you go deeper and deeper into the convolutional neural network, the abstraction gets higher. So these edges are then combined to form angles, and then you put those together, and then you start forming houses or chairs or faces, and then so it's more and more. And this is something very similar that happens in the visual system. So it's yeah, I think there's some parallels. I don't know. I'm not like doing this type of research. There are people doing research where they're trying to bring them closer together and trying to learn, like as a way to develop deep learning. But yeah, there's a lot. I think there's a lot of parallels.
That's all the time we have for questions. Thank you, Ricardo. So before we head off to the next poster session, we have a few more presentations from industry sponsors. So we have 3 5-minute presentations. The sponsors presenting this time are Charles River Laboratories, BioLegend, and LaserGlow. So please welcome them.
Thank you. It's HBHL, I think, if I remember correctly. So my name is Harun Rashid. I work as a principal scientist at Charles River, Montreal. So I remember, I think, giving a talk at this very place when I was a postdoctoral fellow at McGill Pain Center back in 2006, 7, and 8. So maybe some of you may know, but my professor, he already left, Professor Fernando Cervero. I was at the McIntyre building at that time. Anyway, so Charles River. What is Charles River? People are like, what is Charles River? So I say, it's a river. It's a river in Boston. So the company's name actually based on Charles River, actually. It was founded by Dr. Foster, who actually started the animal breeding business 60, 70 years ago. But right now, it's no more the animal supplier. Only 20% of our business is animal supply or breeding. The 80% is drug discovery and drug development. So what is drug discovery and drug development? That is also something I think most of the people in academia are not much aware. So drug discovery, it's called a continuum of drug discovery development. So it's discovery has two main phases. Early discovery, where you identify the target, it's called target identification. Then you do hit identification. Then you do lead identification. Then you do lead optimization. So once the lead optimization, up to that phase is called early discovery. And after that, during the lead optimization, what you do is you do efficacy testing of your molecule in different disease models or animal models based on your intended therapeutic indication. So that's the discovery part. Once the discovery part is done, that you have a molecule that is working in a certain disease model, then you go to the development phase. And for the development phase also, there are two parts. One is preclinical development and then clinical development. So preclinical development is safety assessment or toxicology assessment, and the clinical development definitely. And after that one is done, then you submit for IND, and then you go to a clinic for phase 1, phase 2, phase 3, and then after you do NDA, all those things. So basically what Charles River does is we do early discovery up to preclinical development. We don't do any clinical works. So this is just from this morning. I just copied from our website, external website, just what we do. You see that it's not just animal supply. We do from animal supply in research model to drug discovery to development in different disease areas. I cannot see properly, but you can see, and you can imagine that what we do actually. It's right now we are the biggest contract research organizations in the world, providing services in drug discovery and development. 80% of the drugs last year, that was approved by the FDA. One way or another way, we have been working with those drugs. So in Canada, there are 3 facilities, Charles River facilities, one in Montreal, Saint-Ville, another one in Sherbrooke, and then we have a breeding site at Saint-Constant. Montreal facility is one of the biggest facilities among the Charles River organization. We have about 1,700 people working in the Saint-Ville facility, 300 people at Sherbrooke, and about 200 people in Saint-Constant. So we are one of the biggest employers also, top employers of Montreal. Last 2 years we have been. In terms of drug discovery expertise, as I told, so we have been working from discovery to development, but I'm just going to focus on the drug discovery part because I work in the drug discovery field. So I work at the Saint-Ville site. Although the Saint-Ville site is mostly for toxicological assessment, but we also have a little small group that does drug discovery works, mostly in IBD, dermatology, pain, inflammation, those kind of disease areas that we do. So basically every disease area we cover, and from early discovery to up to the safety toxicology assessment we cover. This is the discovery services that we provide at our Montreal site in pain, arthritis, IBD, different immunology models, respiratory models. So Montreal site is called a Center of Excellence for Inflammation. Just wanted to show something, that we use for assessing pain in animal. That's all. And this is just an image, just to just imagine what it is, if you can, any of you. Thank you. Thank you.
Awesome. Hi. So my name is Karen Morley. I'm an application scientist with BioLegend. And today, in my 5 minutes, I'll give you a quick overview of our neuroscience portfolio. And then I'll quickly tell you about a new product line that we're super excited about. So BioLegend is a leading provider of antibodies, biofunctional proteins, and immunoassays for research in immunology. That's what we started out in. Cancer, cell biology, stem cells, and of course neuroscience. We have over 18,000 products in our catalog to date, and we've been cited in over 27,000 peer-reviewed publications, and we're still privately owned. So our core values are world-class quality, superior customer support, and outstanding value. So that's at the center of everything that we do. And down in the corner here is a picture of the new BioLegend campus. It's at BioLegend Way, and we've been in there for about a month now. So this is a schematic of product development at BioLegend. So this is particularly for our antibodies, and it's just to show that all of our product development, our validation, our quality control is governed by an ISO certification that ensures that we are developing sound quality products and for you guys to use in your research. So this is a product overview. As I said, we make antibodies. We also do cytokines, chemokines, growth factors, so your functional proteins. We do immunoassays. We do chemical probes, and we do neuroscience reagents. So this is sort of, some categorization of our neuroscience portfolio. So it's usable in areas like neurodegeneration, neurodevelopment, and basic neurobiology. And some of the diseases that we focus on in our portfolio is Alzheimer's and Parkinson's. This is a bit more categorization of our portfolio. So there's protein aggregation, antibodies as cell markers. There's neuroinflammation, synaptic biology, and protein trafficking and clearance. Some of the top techniques that our reagents are used for are Western blots, immunohistochemistry, immunofluorescence, with some use for flow cytometry and ELISAs. And this is just some examples of some of the targets that we offer as antibodies or, 2 minutes left, as antibodies, ELISAs, or recombinant proteins. And it's by no means an exhaustive list. It's just some examples. So switching gears a little bit, I wanted to quickly tell you about a technique that we're super excited about. So typically when you're doing research and you're looking at DNA, RNA, or protein, you're doing analysis, you use one technique for your RNA expression, you use another technique for your protein expression. So I'm going to tell you about a technique that combines more than one. So it's a multi-omic approach that combines proteomics with transcriptomics, and it's a technique called site-seq. And we are we're well into putting out a nicely developed portfolio for these antibodies. So CiteSeq, it's the cellular indexing of transcriptomes and epitopes by sequencing. It was developed out of the New York Genome Center back in 2017, and it uses single-cell RNA sequencing to simultaneously detect cell surface markers and transcriptomes of single cells. So BioLegend and the New York Genome Center entered into agreement for this technique back in 2017. And this is the total-seq antibody. It is how you're able to get a sequencing readout, get a protein expression from a sequencing experiment. And if you want more information about the antibodies, please come by the table and we can talk more about it. So this is a quick overview of the workflow for CiteSeq. So it goes from incubating your cells with the total-seq antibodies, partitioning your cells into droplets with beads, cell lysis, your mRNA and oligo hybridization, reverse transcription to get cDNA, PCR amplification, library prep, and then sequencing. So we provide the antibodies, so the total-seq antibodies, and then everything else up to the sequencing. 10x Genomics is here. You can go by the table. They have kits and instrumentation that allows you to do the bulk of this workflow. And then also Genome Quebec is here as well, and they house instrumentation and they provide services that would allow you to do this whole CiteSeq technique. So this is my contact information. Please, if you have questions, stop by the table, ask me, send me an email, phone me. I'm happy to help in any way I can. Thank you so much for your attention.
Just using it for notes. Hi everyone, my name is Nick. I'm a technical sales representative from Laser Glow Technologies in Toronto, Canada. We're a small business. We're an optoelectronics company specializing in the sale of laser systems. Laser Glow originally started off as a small company supplying to the educational and hobbyist markets. Demand quickly grew as the industrial and scientific markets for laser equipment established Laser Glow as a recognized player in this market since about 2005. One of the largest uses of our lasers is within neuroscience, specifically optogenetics. As many here will know, optogenetics is a method using both genetic and optical technologies to activate and deactivate specific cellular behaviors with high spatial and temporal resolution. Laser Glow's lasers are widely used in optogenetics, both because lasers are coherent and monochromatic sources of light, meaning that a very narrow wavelength can be specified and the light output will always be in phase. Laser Glow's lasers can be efficiently coupled to optical fibers, allowing for deeper brain structure manipulation with implants. In fact, our lasers have been used among the pioneering labs, researchers at Stanford and elsewhere in California that popularized optogenetics, and as researchers branched out and established their own labs across the globe, our laser systems have been following them along ever since. Laser Glow also collaborates with academic and research organizations globally, working with research and development, customization of our existing products, and building complete solutions. For example, one of our flagship products, our laser systems, is our Cyanea. It's a 470 nanometer diode system built specifically to address some of the modulation concerns that research had with the modulation performance, more commonly had with the 473 nanometer diode pump solid state systems. So working with academics labs here actually in Quebec, in Montreal, Laser Glow helped in design a manufacturer's system while being tested, reviewed, and used in active research. So Laser Glow could provide the best product for not only the one lab, but for the whole neuroscience and optogenetics fields. On this note, Laser Glow is looking to the future and is collaborating and planning to design and launch new systems for neuroscience and optogenetics, focusing on more compact and full-featured turnkey solutions, making it even easier for new labs to start their research quicker. We're currently fostering many new labs across Canada, US, Europe, providing extra support, access to accessories, enhanced discounts to these new labs to make it easier to source equipment and not just waste time shopping around. Not limited to just neuroscience, we have our systems being used in a multitude of other fields, including microscopy, spectroscopy, particle image velocimetry, and more. Outside of the academic field, Laser Glow also has a strong social standing due to various contributions within our local community and international global village, actively participating with charities and organizations such as Kiva, FoodShare, Stephen Lewis Foundation, Red Cross, as well as programs, participation in programs to help a strong environmental agenda. Laser Glow being based locally here in Canada, we're able to provide real-time support to customers when needed. So if anyone has any questions or applications where you might be using lasers in the future, feel free to stop by my booth or just give us an email or a call. Thank you very much.
All right, before we head off to our second poster session, I just had a few things I wanted to say. First, for you oral presenters from this session, you might have noticed we didn't all get together and take a picture. That's because you're all not here. So it'd be really great if you can come at the end of the closing remarks. If you can meet in this corner here to take a picture together, that would be really wonderful. So keep that in mind. And then a reminder for those who signed up for scientific speed networking, that starts at 4:15. So please head over to the speed networking rooms at that time, as shown on the program. And for those of you who are not at speed networking, please know that wine will be served at 4:15. And for your speed networkers, wine will be brought to the speed networking room, so don't worry about that. And then eventually at 5:15 p.m., we'll meet back here for best presentation prizes, door prizes. So make sure to get those stamps if you haven't already, and the closing remarks. So I'll see you back here at 5:15. Thank you. Oh, and also, judges, poster and presentation judges, don't forget to hand your judging sheets at the end of this next session at the registration table. Well, thank you.